see. Okay, the job has done, I think. If you want to see the job status, you can go to run and see the job status. So it's done. It's not here. So let's see that it has either created the DLG file or not. So let me go to my folder of the docking, and here you can see that these files, these are formed. So I got these doc.dlg file. Basically, this is a docking log file. And here you can see that it's just finished at 6:13 p.m. So it's just finished. How we can analyze that? So first we see that what is in that particular file actually. If I right click on that, I will open it into the VS Code. And in VS Code, it will ask me, do you really want to open it? Yes, I want to open it. And then here you can see it like this, that in that particular file, it will tell you this is the AutoDock 4.2 file. And these are the parameters which you usually use. These for the map files which you are using for these things. And these are the ligand molecule PDBQT in which different charges and different things were present. And then it will just let me know that it has started genetic algorithm from here. Final value was minus 7.0, and this is the run one. And then after that, it has just sorted the things. This is run 2 out of 50, and this is the value. So I will go in the bottom now, and in the bottom here, you can see that it has just completed its run somewhere here, and it's just letting me know that what was the energy values. So in the bottom, if you go, then here you can see different things, different parameters that in every run, what was actually the real energy. So this is the CPU time, and this is system times, and this is the real time, means in 6 minutes, this file has been run. So when you run the larger system, then it may take you for about 10 minutes, 20 minutes, or maybe 60 minutes, depending on the atoms which are present into your computer. So this is how your DLG docking log file that look like. So how you can analyze it? You can go to analysis file, and here is an option of the docking, and it will ask you want to open the docking logs. I will say okay, and here is the doc file. I will just open it, and here it will say that how you wanna give the ligand name. I will say it ligand 2, and then I will say it okay. So it will ask it read 50 doc confirmation from doc. We got the 50 ones. You can analyze by confirmation and play. I will say okay. I will go to again analyze the confirmation and play. So here are different options: play or play by rank by energy. I will say it play, and then here what I want to do, I want to color this ligand to display actually where it is. I want to see here it is, this one. And if you want to color it by different atom types or different color, you can color it. I will give it a custom color. I will ask it, I want it red, and then I will just put it like this. So I will close it. So this is my ligand. Now let's see that in which conformation it's usually found. Here you can see that how it's moving actually from one position to another. So here you can see, or here you can see it one by one actually, that which conformation you want. This is the 50th one, and this is the other ones. You can have a look at that. So this is how you can see where the ligand is particularly binding. You can color it in a different way, and then you can check it. So these are the few options. So this is an option panel, and in this optional panel, even you can see that show the information about this particular pose. The binding energy is minus 3.0. This is ligand efficiency. This is ligand constant. And these are the internal energy, electrostatic energy, and other things which are present here. I have made some script which will just extract all of these things to make a table actually. But later on, here you can also build the box actually, but you need to give it your ligand, your molecule, which is the macromolecule. How you will do it? You will go to macromolecule and then you will ask it to choose. So this was our macromolecule. And now if you want to make the hydrogen bonds, then you can just go here and it will make the hydrogen bond. It says hydrogen bond is present between salmon, glutamine, third hydrogen, and the leucine, which is present at 92 number, it's HN. So hydrogen bond is present there. It will show the distances actually. Actually, we are not going to analyze our docking file on AutoDock because it's bit complicated and it's pi mole. It's very easy, but you can get these options actually from here. But one thing which you need to do is to write complex. Yeah, when you will say it, okay, I will just go to the top energy complex actually. I will not go here. So now it will give everything info. I don't want info for everyone. I will just go to the compound which is of the best energy and which is present at top one. I will say it right, complex, and I will give name it as complex. So here are certain options. You can write it down as PDBQ. So it has written the file in form of PDBQ. So I will just stop this. I will stop it. And then if you want to play the confirmation, you can do it. If you want to do the clustering thing, you can even cluster it. So in the clustering, you can see that what is the RMST and binding energies actually. So this is RMST, that how many poses of this energy are present and how many poses of this energy are present, and this is the binding energy. So here you can see that how binding energies, these can be analyzed. You can make this graph by yourself by extracting the information or clustering, reclustering and everything. But ultimately, what will happen if you go to the docking? Here we got a complex. So this complex is .pdbq actually. So if the name is not clear, you can say it pdbq. So PDBQ file has been written now. And now this PDBQ file, you can open it into PyMol if you want to. And in PyMol, actually what you will do, you will change the PDBQ into the PDB file. And you can also do it by using this AutoDock tool, that you will just delete all of these things which are present here because you don't need now, and everything has been done. You will go to delete and you will ask it delete molecules. Here you can select all of these and you will ask it delete molecule. So oh, here is another error. So there is some error in actually in some library that this delete command is not properly working. So it happens usually. So let's read it. I don't know now it will read it or not, or it will just stop working again. But let's see. So here is again the error. We need to restart it. I'm not going to do it. So what you can do in PyMol, you can open that particular file and you will ask it to read. So I think on desktop, I have put that. So you will just, you can also check the maps here even. So maps, they are usually open in that particular thing. So I think it was PDBQT because QT has the charge, or it hasn't actually read the molecule. So it's not reading it here. So it must be PDBQT, in which format it's getting saved. So I need to change its name. Rename. I will just rename it as such. Okay. I will not gonna rename it by extension. So I will just gonna rename it as such. So let me rename it as complex. Okay. Yes, I wanna change it. Okay, let me then open it because I wanna open it as such that if it's open it or not, it's not opening it. It's saying that it's unsupported file because I think this is again some error in saving this file. I will say it all files, and I wanna, because it should open the file because it has saved it into that particular format. So here you can see that it has opened it, but it says that it has not been recognized. Which format you wanna use? I will use PDBQT, I think. And in PDBQT here, you can see that this AutoDock complex has been opened. So I will go to File, and here files, there are again several options. I will go to Save, and I will ask it to write PDB. So this is complex.pdb now. I will say it, okay. And then I can open this particular PDB. It will be somewhere where it is. It has saved, I think, here. Let me, here. And I will open it. So it has been opened in PyMol. So in PyMol, we can see it here, and even we can see the ligand here, which is particularly present in this location, in this form. So how we can get ready our publication ready figures here? I will, very simple thing, I will go to action, and here are certain presets which I have showed you. You will go to preset, you will go to ligand site, and you will ask it to cartoon. And here you can see that it has given you that how the ligand molecules actually interacting with these things. Okay, what are these? These are actually the missing residues which were present between the chains. Either you can build that beforehand with homology modeling if your ligand is present very near to that, but if you don't need to build that, then it's quite fine. You can just survive with that. And then what you need to do, you need to select. So I think internet was some issue. So you can see that you need to select all these residues, these amino acids which are surrounding our ligand molecule. You will select that, and after selecting that, you can label that actually. Usually use the mouse, I think, for that purpose, because even I am unable to use this on this laptop actually, on the Windows one. So here is an option. I will say it label. Label what? Label residue. And here you can see that it has labeled all the amino acids which are surrounding that particular ligand. And then we can check with the literature that either this is the ligand which we require or not. I will send you the file, and here is the active site is included in that. And then you can check that either these tyrosine molecules, these are the actually involved in the process of interacting with the ligand molecules or not. So this is how the ligand molecules they are checked, and if there is any hydrogen bond is present, that look like this. Like here is a hydrogen bond is getting formed, and this shows the polar contact. If you want to hide it, if you want to display it, you can do like this. So this is how these ligand molecule is now bound here, and if you want to make the picture of that, then what you will do, you will go to the ray thing, and then you can copy paste or you can save the molecule. And I will just save the image file. I will save it as a 123, and then if I go to my docking folder, here you can see that my image file is here, which is in name of 123, and this is my image which is present here. But you people still need to refine it, because some names, they are present under the amino acids. So what you need to do, you will do that, you can press the control button actually, and I will just put it a big here. So what you need to do here, an option, the viewing option and editing option. Now you want to edit something, you will go to edit, and then you will press the control, and then by pressing the mouse button, you can just move these residues to the certain spaces where these are required. So this is how you can adjust. But remember, don't touch these atoms, otherwise you will also touch and remove these atoms and move them. Don't move them, just move the names of these amino acids. And then after removing the names of amino acids, then you can again ray the diagram. So I will also let you the ways that how you can use these commands on the command prompt of the PyMol, and then you can easily do all these kind of things and manipulate and generate the figures actually for your uh, papers and the project. So this is how you analyze one thing. And then the second program, which I will send you the links, and that is actually called the ligplot. Ligplot is another program by which you can open and check the PDB files actually, that how the ligands they are interacting with the protein molecules. So I think our folder was name of the docking, and our final file was the complex.pdb. So this is the file, and it will asking me this ligand. Yeah, we got the E2, and I will say it, run it. And here you can see that you got the publication ready figure again, that this is how the ligand molecule that is interacting with that. So you can make its resolution better, and even you can directly link it with the PyMol if you want to. I will make some videos that how you can install and you will analyze these things by using this particular program, and you can analyze these cavities and electrostatic interactions, that which particular ligand portion is interacting with the amino acid and what is their electrostatic links. So you can easily generate them. So this is about the lig plot, that how you can use the lig plot. If you want to increase the sizes of these ligands, increase this thing, I think your computer resolution that should be big, a little big, or here you can also change the parameter size. Actually, you can change the parameter size and then you can write it like this. So this is how the lig plot actually works, and on which you can see these figures. Another way of doing, I will send you the lig plot link and then you can download it. It is somewhere here. I have just downloaded it before the session somewhere, I think, and that's here actually. And here you can download you need to download the Java actually before Lakeplot. So Lakeplot is somewhere here. So Lakeplot is here. You just write down Lakeplot download into Google and it will take you this particular link. I will send you the code, or I will send you just the program in the zip file, and then you can do it. And then second analysis program is basically this Philip, in which you also select your PDB file, and then you will upload it, your PDB file. Once it's uploaded, you will ask it to analyze. And then it will automatically analyze the things. It will let you know that where particularly your hydrogen bond is present, hydrogen bond donor is present, acceptor is present, and that look like this. You can download its results and then you can analyze it in PyMol. So actually this is telling you the information like this in form of the table. These residues which are showing hydrophobic interactions, these residues which are showing the hydrogen bonds. Actually, the Lig plot shows you hydrogen bonds which are lesser than the 10 Da difference. But it shows you more than, less than 3. It shows you more than 3. You can also change Lig plot parameters to look at these distances which are more than 3.3, but you need more time to learn actually, but you can do that. And these are the pi stacking interaction if you want to see that. So all these things, they are reportable. You can write down into your thesis, into your papers. And another thing is UCSF Chimera. Actually, I want you people to download this program, because this program is also very good actually, to see if there is any question. I wanna take the question, I think, today. And which basis we select the ligand. Actually, the basis I told you in PyMol, the PyMol, when you select the amino acid residues are appeared. So I will send you a script actually, and we will do some scripting. Therefore, that was the reason that you people learned about the Ubuntu and the Linux thing, that Linux make it very easier that you can extract the amino acids from the proteins and the ligand interaction site. So it will become very easy. So I will just stop it here. UCSF, I wanna tell you, show you people download it from here, Windows, and that's very good program actually. UCSF Chimera, and then you can search it in Chimera, and Chimera, it will starts like this. And even why I wanna use you people, because it gives you also the docking results, that how you can see the docking results. I think we have made the docking result in form of GLG, but you people need to open your receptor and other things beforehand. So it opens the files for you people. Maybe it will not open. So, but you can try it later on. But after doing the basic things, you can open it. Here you can see it has appeared something, but it says it doesn't like it actually. It is not the doc format, but it has given us something, that these are the ligand molecules actually. And if you have opened your receptor molecule, the ligand molecule will give you certain sites, and even you can see more than one site. So this is how you analyze these kind of poses. So these are the all poses, And if I open actually the same complex file here, I think it was somewhere here, complex.pdb maybe. So these are the ligand molecules which are just interacting at these particular sites in our protein. So, and here you can see that even we can analyze that which particular position our active site is present and which is the preferred one which we want to use. So this is how actually you can check, and then you can see that how the ligand molecules are interacting with the certain proteins or not. But you need to play with these tools, you need to learn them. I just tell you one thing, that you will install it, you will go to tool, you will just open your receptor, you will go to tool, and then there is option of surface and binding analysis, and here you will go to view doc, and after you will see all these DLG files actually, that how these DLG files these are present. So you will just ask it what format it is, and after then you can run it. But it's giving us some error actually about the DLG file, although it's giving me these all ligand molecules, but it also give me here RMST and binding energy here. It extract these things, but it's not extracting it here. Maybe some error or some files which are not completely run. And maybe I'm just closing it. If you people have any question, actually, I wanna take that and we can discuss the things now. So it's open for the questions actually. I think you can turn on your mic and you can ask me the things. So in chat, I think there are certain questions. The basis I told you that how you can analyze that. Yeah, recorded session I will upload on the YouTube. And I will try to make the videos which are a bit the clearer and the shorter ones for these ones, and I will also upload the sessions actually. It didn't start in my laptop. What didn't I have got? I used the Windows 11. So actually what happens, AutoDock 2, AutoDock 4.2 actually, that is very old program. It runs very good at Windows 7 and Windows 10 actually. Windows 11, it got some problem, but the issue is that you can run it either on the command prompt, which I will show you later on, or you will just do the restart thing and restart your computer and do it, because sometimes there is a library conflict between the new libraries and the old ones. Ligand, either we have downloaded from PDB or which one. I have sent you the ligand files. You can extract any of these. Okay, for now you need to practice one ligand, one protein. But later on in the next week, means in the next week sessions, not on Saturday, the next week session, they will be about the more ligands and one receptor, which we will do by using the AutoDock Vina actually, which is called virtual screening. And we are going to do that here. And ligand you can select and download from the PubChem actually. The first, I think, if you remember the databases in one session, we had discussed that how you can download the things. Okay, then if there is no any question, then we will just stop it here. Yeah, we will send you the tutorial timely. I hope so. I have sent you the different files today, and you can practice that till the next session, and then you can ask me if there is an issue. And these links I will send you today. I hope so tonight, and then you can practice that, because I don't have installed my WhatsApp and other things on this laptop. So the copying the things and then emailing the things to the other laptop and then uploading the things from there. So it takes a bit time actually. Otherwise, it's quite fine, because few files they do not talk my Mac laptop actually, the Windows one. So I need to convert few things and then send you people. So anyway, if there is nothing to add, then we can stop it here. And then I hope so. I will send you a practice this Saturday, or I will try then, because acetylcholinesterase you have tried. Now I want you people that you download some random ligands and try them against acetylcholinesterase and send me some screenshot. Don't forget, in the discussion group, and we will see that how beautiful figures and diagrams you people are making. in the interactions of the ligands with the proteins by using the pi-mole and other things. And this lig plot I will send you in the zip form, and then you can use it as I have told you. And I will also make a video for that how you can use the lig plot. Okay. Thank you so much then, and see you on Saturday, same time. See you there then.